Hello all, I am Dr. Gauri Abhyankar, working as Assistant Professor in Abasaheb Garware College, Pune. We are going to study endoplasmic reticulum in this session. We are going to divide our study in two parts. This will form the first part, wherein we are going to primarily focus on structure and functions of rough and smooth endoplasmic reticulum. As you know, endoplasmic reticulum is highly branched and complex network of membranes. Now this network of membranes is formed by folding the membrane to give lamellar appearance, or they are folded in lamellar fashion. These endoplasmic reticulum are present throughout the cytoplasm. Now remember that endoplasmic reticulum is a single membrane system and it is the largest membrane system in most of the plant cells. If we talk about functions of endoplasmic reticulum, it is the major site for membrane biogenesis. It is also very important or crucial in secretory pathway. Besides these functions, it is also required in many different accessory functions. The learning objectives for this session will be: first, to study the subdomains of ER. Second, to study ultrastructure of rough and smooth endoplasmic reticulum. Third is to know about shaping of ER tubules. Next is to find out more about contact of various organelles with ER. And last one is to study the ER quality control system. Although ER is a single continuous compartment, it is composed of structurally and functionally different domains. The ER in interface is divided into two types: nuclear ER and peripheral ER. Nuclear ER is also called as nuclear envelope. Peripheral ER is further classified as rough and smooth endoplasmic reticulum. Many of these subdomains are dynamic in nature, which gives ER remarkable flexibility to alter structural organization so that they can adapt to changing cellular requirements. The ultrastructure of ER, as you can see in the slide, has been visualized by electron microscopy. Rough endoplasmic reticulum is studded with ribosomes, so it appears more granular. It mostly has tubular appearance, whereas smooth endoplasmic reticulum is highly complex than rough endoplasmic reticulum and is more dilated and convoluted. Rough and smooth endoplasmic reticulum show different membrane compositions. Majority of the membrane proteins are same for both, and these are called as general ER proteins. Besides these, rough ER shows abundance of many other proteins that are involved in translocation or processing of newly synthesized proteins. So, rough endoplasmic reticulum are present in all cells because protein translocation is essential for all eukaryotic cells. Smooth ER is present in only certain cell types. Inner nuclear envelope proteins are synthesized on rough endoplasmic reticulum. The classical nuclear localization sequences cannot target membrane proteins to the nuclear envelope. So these proteins most likely travel through the continuous outer nuclear envelope and enter into the inner nuclear envelope membrane by diffusion through the pore membrane. The figure shows you 4 steps for targeting and retention of an inner nuclear membrane protein. The movement of protein is shown by white arrows. First, step A shows translocation or transport of newly translated protein into the rough ER membrane. This protein diffuses through the peripheral ER, which can be observed as step B. In third step, that is step C, the protein diffuses through the nuclear pore membrane into the inner nuclear envelope. Finally, when the proteins arrive in the inner membrane, they get associated with lamins and or chromatin, and so they are retained. The inner nuclear envelope membrane proteins that bind directly to lamins or chromatin are likely to be responsible for establishing and maintaining nuclear envelope structure. Smooth endoplasmic reticulum is generated when the ER surface area required to accommodate resident membrane proteins exceeds than that needed for ER associated protein synthesis. In some tissues, smooth ER separates from rough ER and assumes distinct organization. The most common form of smooth ER is a tubular network with different characters from polygonal meshwork. Smooth ER tubules are more intricate and show frequent branch points. This architecture of smooth ER is called as random tubular. Smooth ER forms parallel arrays of stacked cisternae in those tissues which overexpress ER membrane proteins. There are many types of stacked ER and they are collectively called as organized smooth endoplasmic reticulum or OSER. Different forms of such organized smooth endoplasmic reticulum can be observed in this slide. If the stacks are arranged against nuclear envelope, they are called as carmeli, which are observed in figure A. Here N indicates the nuclei. Also, at the distal side of the carmela is a forming cisterna that appears to be generated by the fusion of tubules. Figure B shows stacks of endoplasmic reticulum that are distributed elsewhere in cytoplasm but not against nuclear envelope, and they are called as lamellae. The arrows indicate continuity with the cytosolic space. It is important to note that the width of the space present in all OSER structures is constant and it corresponds to the cytosolic space bridging the cisternae. Figure C shows formation of concentric whorls. Note that the whorl is in close association with the mitochondria. Figure D and E show regular sinusoidal arrays with cubical symmetry. In such cases, they are referred to as crystalloid ER. In figure E, the asterisk indicates the luminal space. High expression of certain enzymes like HMG coenzyme A reductase induces tubules to form bundles with hexagonal symmetry that is shown in figure F. In Arabidopsis, another type of ER structure has been identified that is known as ER body or fusiform body. They were observed in addition to ER network as novel rod-shaped structures. ER body is about 10 microns long and 1 micron wide, and it is the largest ER structure in plants. They are surrounded by a single membrane and are attached to ribosomes. Also, they are connected to endoplasmic reticulum, so they are considered as subdomain of ER. These ER bodies are filled with condensed material, so their specific gravity is higher than that of endoplasmic reticulum. Dry seeds do not have ER bodies, but ER bodies are accumulated in cotyledons after 4 days of seed imbibitions. The figure also shows you ER bodies in cotyledons of Arabidopsis. These ER bodies disappear slowly with progression of senescence. It is also observed that the number of ER bodies increases when plants are wounded. Usually, the accumulation of ER bodies is slow and requires 48 to 66 hours. Peripheral ER is enriched in proteins that shape membranes into tubules. Two classes of proteins, that is reticulon and DP1 UP1, are involved in shaping the network of ER tubules. Both these are highly conserved integral membrane proteins. The slide shows schematic diagram of the tubular endoplasmic reticulum. Reticulon family of proteins contribute to bending of membrane by forming a transmembrane hairpin topology. This structure acts as a wedge which moves lipids in the outer leaflet of bilayer to some other position and ultimately leads to membrane curvature. If some reticulon isoforms are overexpressed, there is formation of long ER tubules at the expense of sheets. Alternatively, depletion of reticulons reduces the ability of membranes to bend. Due to this, there is reduction in number of ER tubules, leading to expansion of ER sheets. So, level of reticulons in a cell governs the abundance and fine structure of ER tubules. Along with reticulons, members of DP1 or UP1 or REEP5-6 and REEP1 to 4 family also act as tubule promoting factors. These are ER resident proteins. They are present in tubules and also at the edges of sheets. ER quality control system mediates and monitors the processing and folding of secretory proteins destined for transport to the plasma membrane and organelles. It identifies misfolded proteins and transfers them to the ER associated degradation machinery. Plant ER processes many plasma membrane resident proteins involved in adaptation to environmental stress. The slide shows signaling in plants under mild and prolonged ER stress. Models indicate overlaps and differences in ER stress signaling. Under stress conditions, any disturbance in ER function can result in disturbed plant development and plant immunity. Environmental stresses create an increased requirement for secreted proteins. If this demand exceeds the ER quality control working capacity, unfolded proteins accumulate in ER, and this condition is sensed by cells as ER stress. Prolonged ER stress impairs ER function and so threatens cellular integrity. But these models to study ER stress need some gaps to be still filled. You can see various causes of unfolded protein response and ER stress in the slide. Whenever there is overload of unfolded or misfolded protein in ER lumen, the condition is called as ER stress. Disturbances of ER homeostasis cause ER stress that triggers the unfolded protein response. This response may be induced by pharmacological chemicals, reductive or oxidative agents, as well as non-steroidal anti-inflammatory agents which impose stress on ER by causing vigorous protein synthesis, imbalance of ER calcium and redox. It also inhibits protein modification or transfer to the Golgi body. ER stress also occurs under many circumstances such as nutrient deprivation, developmental processes, genetic mutation, as well as pathogenic attack. Recent reports in plants have indicated a close connection between the UPR and environmental stimuli such as heat, salt and drought stress, as well as viral attack, but the underlying mechanisms are largely unknown yet. This is IRE signaling pathway in plants. Arabidopsis possesses at least two IRE1 like proteins, ATIRE1A, ATIRE1B and OSIRE1 harbor all structural features of yeast and mammalian IRE1. ATIRE1A and OSIRE1 are capable of autotransphosphorylation, and the putative ER stress sensor domain of ATIRE1A, 1B and OSIRE1 can functionally replace yeast IRE. There are at least 3 ER resident transmembrane B-zip transcription factors in Arabidopsis, namely AT-BZIP17, AT-BZIP28 and AT-BZIP60, which are involved in ER stress responses. The unfolded protein response or UPR is supposed to ensure cell survival. The molecular basis of plant ER PCD and the role of plant BZIPs in signaling are largely unknown. Now let us summarize what we have done in this session. First, we studied the structure of rough and smooth endoplasmic reticulum. After that, we have studied the tethering of inner nuclear membrane proteins, that how these proteins would move from endoplasmic reticulum to the nucleus. Later on, we have looked at ER bodies. We have seen their ultrastructure and their functions. Then we have seen how the membrane proteins or how these ER membranes are tethered and how these ER tubules are formed. And finally, we have seen or we have looked at ER stress, then unfolded protein response and the signaling pathway for the same.